Hello guys, how are you all doing? I hope uh, throughout the course you had a good time so far, you're enjoying the course, and if there is something that I can do to help out, please let me know. Okay, so last time I talked about these guys, the embryology. How come my things are not multipotent and totipotent? And then this is a chick, 33 hours chick embryo, the diagrams of it, that all of these parts in here you should know. And also that is serial section. It means they took the animal and they cut it serially. I hope you know what I mean. So the very first top part is from the head region, and this is from the heart region, and this is from the somite from here. So from the head, from the heart, and then from somites lower down here, and toward the end of the animal, toward the posterior end of the animal, toward the tail. That's where you have it. Okay, and then of course this is the whole entire animal, the whole mouth. So what happens in a microscopic slide, you know, if you look at it in the lab, of course we do not have lab now, but if you look at something, it says WM ... It means the whole animal, the whole organism is in there. But the last one, you will see, you know, chick 33 hours, and it says S serial section. Okay, SC or sometimes serial section, SS, I'm sorry, SS serial section. And then if you have something that you're like the bone, you're looking at it, then you will see cross section, CS. Okay, that's where pretty much if you see those abbreviations next to the slide or something, that is talking about, you know, how the slide was prepared. Was it serially cut just like that one? Was it just my finger? They cut it right here, and you're looking at it from top, okay, or you're looking at the whole entire organism on the microscope. Okay, so the next thing, let's talk about Protista. But before I go to talk about Protista, there are a few small little things that I need to talk about and settle over. And one of them is that the you know, Robert Whittaker, it is in your notes, his spelling of his name and everything. In late 60s, he proposed this. He said, okay, prokaryotic cells evolved to Protista, and Protista then evolved to some of the Protista evolved to animals. Some of them evolved to plants. Some of them evolved to fungi. And that's what during exam, I say animals evolved to fungi, fungi evolved to plant. That's wrong. That did not happen, and no scientists believe that. They believe that Protista evolved, some of them, again, evolved to plants, some of them evolved to fungi, some of them evolved to animals. Okay, so they're kingdom of their own, fungi, plant, animal. You learn that in Bio 1. So they are the kingdom of their own, and one did not evolve to another one. And then, of course, the prokaryotic cells, you know, the cells that they do not have nucleus, like bacteria, archaebacteria, the two domains you learned in Bio 1, and the archaea and bacteria, there are two domains, and the other one, eukaryote domain. In the eukaryote domain, you have plants, animals, fungi. So those are, and Protista, of course, they are all in the domain eukaryote. Okay, so let's move on. And then the classification of all living organisms on planet Earth, there are about 1.5 million. That number has changed. It's changing constantly. I would say it's about 2 million. It's about 1 million, just 1 million arthropods have been identified, and there are being more identified. But anyhow, so let's say I'm not going to ask you that number. There are 1.5 million species that have been identified so far. Let's assume that. And then out of that 1.5 million, a good number of it are animals. And, you know, as I said, arthropods are about 1 million. Carlos Linnaeus, he was a medical doctor, as far as I know, based on what I read and I remember. That's the thing, based on what I read. And then he said, this is chaotic. Let's put organize things, the name of the organisms. Let's organize them. So if I travel to Japan and I say Homo sapiens, they know what I'm talking about. So all of the scientific community around the world use the same name for the same organism. And that's what he proposed. And then of course he proposed there should be two names, like Homo sapiens for human. I'm saying Homo sapiens, so you know, for rest of semester, you're going to learn a lot of scientific names. So binomial nomenclature, it means two names, Homo sapiens, and then based on comparative anatomy. That is changing a little bit now. They use anatomy, but they use a lot of DNA nowadays to put organisms in. King Paul came over for good sakes, so we'll talk about that in a minute. Here we go. Speaking of devil, King Paul came over for good sakes. So that method of classification still being used a little bit, still is being used, but we are having nowadays clade and grade, and I will talk about them, but I don't know if really you should know them, but we're sticking to this. King Paul came over for good sakes, and that's in that order. King, kingdom, phylum, class, order, family, genus, species. Okay, and then throughout the semester, of course, you know the kingdom. We are talking about kingdom Animalia, and then phylum. You're going to learn, I would say, 30 phylum or so, something like that. Class, we'll talk about in each phylum. There are classes that we talk about them. You should know them. And then order, when we get to arthropods, then the name of the orders you should know. The rest of semester, do not worry about the order. Family, no, nothing you have to know, just a couple of things here and gather, like feline family, I will talk about it. Canine, feline, those are a couple of families, but we will not talk about it. And a lot of genus and species, you're going to learn the rest of semester. How to write a scientific name. First word is written with capital initial letter. Second word is species, and it is all in lowercase. And both words are italicized or underlined. So when you write in handwriting, you must underline it, okay? But when you type, you can either underline it or italicize it. Most people italicize it, but anyhow, example. Homo sapiens, that line, I'm sorry, that line should not be here. I thought I got rid of them, but I guess. Okay, so the line between Homo and sapiens should not be there. Homo underline, sapien underline. And then it can be italicized or gorilla. This line, that line should not be there, but anyhow, or you can write down gorilla. That scientific name for gorilla is gorilla. Name of genus, species. The genus is gorilla, species is gorilla. Then sometimes you will see a scientific name with three names, like this one, Trypanosoma brucei rhodensiense, Trypanosoma brucei gambiense, Trypanosoma brucei. What does that mean? It means the species have different strains. They look alike very much, but they are, the DNA is different, and the region they are from is different. They cause different disease, okay? So, but they are same genus species, okay? So they call these strains, if you would, right here. That's a name of strain. And this is the genus, and this is the species, right? You all know what I'm talking about. And then in sentences, you might see this Trypanosoma sp. It means species. It's referring to all three of them, or all four of them. There's another one, Trypanosoma cruzi. Gorilla can be written down as G. Of course, you always have to spell out the whole word, not world. You have to spell out the whole word, and then you can write down capital G dot gorilla, okay? Or Trypanosoma T dot brucei. I hope I'm making some sense. Okay, Trichomonas vaginalis, you can write it on T vaginalis or T vaginalis. So, but you have to spell it out first time. You have to spell it out completely. And then phylogeny, you know, classification is based on homology, not analogy. If a bird can fly and a bat can fly, they both are not in the same group. Okay, a mosquito can fly and a bird can fly. That's analogy. Analogy means they both can fly, but they're not in the same group. They're not in the same class or phylum sometimes. Okay, so phylogeny is based on homology. Yeah, my arm is similar to bat's wing, so we are in the same we are in the same class. Okay, same phylum. Human and Robert Whittaker, I will, the human, you should know the classification of human, and I will present it to you. Robert Whittaker already talked about it, so there's nothing there. So human, right here is the name of, if you would, so in the animal kingdom, if we have 33 phylum, phyla, plural, phylum, singular, phyla, if you have 33 phyla, so one of them is chordata. So we human belongs to phylum chordata out of the 33. And then do not worry about subphyla. Subphyla is tetrapoda, it means four. Tetra, it means four. And then class, you should worry about that. Human is in class Mammalia. And subclass, again, do not worry about it, Thera. And then order, primates, definitely you've heard about primates, the monkeys, they're primates. And then families, Homondiae, genus is singular, genera is plural, phylum is singular, phyla is plural, genus is singular, genera is plural. So there are 3 genera in Homondiae. Out of those 3, the only survivals are us, human, Homo, and the other 2 are extinct. And then species, we belong to the sapiens, and the other 2 species in that genus Homo, they're extinct. Okay. Okay, the animal like Protista. So all of the introduction I had to give you about evolution, a little bit of evolution, and also the classification is over. So let's talk about animal like Protista. So the animal like the Protista, first life on Earth was 3.5 billion years ago. Now that number constantly changes. So do not, again, worry about that number, but have some rough idea. Now, the most recent one I heard in the news, it was 7 billion years ago. The first life was on Earth 7 billion years ago. Again, those numbers change. And I don't expect you to know that, but you should have some ideas how long ago life was formed on planet Earth. It was very, very long. Protista, kingdom, subkingdom, protozoa, kingdom Protista, animal-like because they do not have cell wall. In some way, they are motile. They do move around, not all of them, of course, and they ingest their food. So when they ingest their food, they are animal-like. And they are plant-like. I did not write it down here. You should know they are plant-like. I would say 200 years ago for sure. Now I'm aging a little bit. So yeah, I would say for sure 150, 200 years ago, all of the organisms on planet Earth were broken down to 2 groups, animal and plants. There was nothing in between. Okay, now with technology and microscope and everything else, they say, oh no, there are bacteria, there are Protista, and they are not like animals and plants. So anyhow, plant-like because some of them have cellulose, some of them do photosynthesis, okay? Some of them they are stationary, they are not motile, we call them sessile. Okay, so these are some of the characteristics that they are plant-like, and I've listed why they are animal-like. So here you go. We will discuss these phyla. I did not list them for you out; they disappeared. But anyhow, you will see. You will see what I will discuss. Some of the characteristics, they are unicellular. They are single cell. Most of them are unicellular. And then they are microscopic. They have no germ layers. You learned that in just last lecture, right? Germ layers, ectoderm, endoderm, mesoderm. They do not have that, okay? And specialized organelles. Some of them have, you will see it again, they have specialized organelles like apical complex in the phylum Apicomplexa. You will see that. Not your ordinary mitochondria and Golgi apparatus and nucleus. No, they have some other specialized organelles that other organisms on planet Earth, they do not have. Anyhow, free living, mutualism, commensalism, and parasitism. Some of them are free living, some of them are mutualism. Mutualism, it means if let me give you the definition of symbiosis. Symbiosis means two different species. It has to be two different species. Like a pregnant woman and a baby, that's not symbiosis. Okay, they are same species, right? I hope I'm making some sense. So symbiosis is two different species that one of them is benefiting metabolically from the other one. That is symbiosis. Sym, it means together, biosis, living, life. Okay, so symbiosis, two different species that one of them metabolically depend on the other one is called symbiosis. Okay, so there are three levels of symbiosis. Mutualism, both benefit. Commensalism, okay, I'm not going to spell it out. One benefit, the other one is not harm. I just put it as a multiple sign of multiplication. And the other one, the last one, which we are interested in this course, parasitism, one benefits and the other one is definitely harm. Okay, so parasitism, and of course, not everything in biology is clear cut, black and white. We have a lot of gray area in biology. That's what makes biology so interesting. Okay, so before I go on, but if you if somebody asks you what is the evolution of these things, which came first, which came second, which came third, now present, now time, what would you say if this is a timetable, if you would? And then at the beginning of the time, it would be parasitism. The animals, one group of animal were metabolically depending on the other animal, and the host animal would die. If the host animal dies, of course, the parasite will die too. Then over a period of time, let's say that was 1 billion years ago, over a period of time, the two organisms learned that they should not harm their host, and that became commensalism. That would be like 500 million years ago. And nowadays, it's mutualism. Not only they don't harm them, but they benefit them. So they can live longer. When the host lives longer, the organism, the other symbiont, I cannot say parasite, the other symbiont on it or in it, whatever, can live longer also. That's called mutualism. They both benefit. Okay, Let's see what happens. Some sessile and some have pseudopods, some have flagella. That's the mode of locomotion. In all types of nutrition, autotrophic, heterotrophic, and aquatic and terrestrial, asexual and sexual reproductions, all of them. They have, if you would, they have most of the organelles that other organisms they have, eukaryotes they have. Come on, let's go. Mitochondria, please make sure you know what they are. Mitochondria is a power plant of the cell, and it provides ATP. In biology 1, we harped on it a lot. Mitochondria provides ATP. Cellular respiration occurs in mitochondria. In the plasmic reticulum, you have two of them, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum, you learned it in the last exam material, like sarcoplasmic reticulum, it stores calcium for muscle contraction. Also, another function of endoplasmic reticulum, in other cells of our body, they make fat, okay, lipid synthesis is a factory for lipid. And a rough endoplasmic reticulum is for protein synthesis, the ones that have ribosomes. Okay, quickly. Golgi apparatus is a packaging part of the cell. Vesicles, they're little phospholipid layer, and they move either fluid or they move molecules within the cell. Okay, I don't want to talk more than an hour. Chloroplasts, they do photosynthesis. As I said, some of the organisms in this phylum have chloroplasts. In Bio 1, you learn chloroplasts where photosynthesis takes place. Vacuoles, some of the species have vacuoles. They store waste material, they store water, they store food, nutrients. That's what the function of vacuole is. Ectoplasm and endoplasm, do not confuse them with ectoderm and endoderm, please. Ectoplasm and endoplasm, they are responsible for locomotion of the pseudopod. Okay, so pseudo. Locomotion, speaking of locomotion, Bio 1 again, you learned 9 microtubules in the parameter, 2 microtubules in the center, that is 9 plus 2, which is found in cilia and flagella. Pseudopod is false foot, literally the definition, they move, amoeba move by pseudopod, and paramecium sperm move by flagella and cilia. Ectoplasm, transparent or hyaline colloid, and endoplasm, more granular, more fluid. Okay, this is what you learned. This is a transmission electron micrograph of a flagella or cilia right here. You can see 9 microtubules in the parameter right here and 2 in the center. So that's where you get 9 plus 2, okay? And then of course, it's surrounded by cell membrane. This is cell membrane. That's what one of the questions that I like to ask during Bio 1. What is 9 plus 2? The microtubules are surrounded by phospholipid bilayer. They're surrounded by cell membrane. And the way they move, if you take a course later on, cell biology or histology, you learn that they move just like muscle contraction. Remember in this class you learned your muscles move, they move just like that. Okay, and we don't go into details of it for this course. Okay, here is ectoplasm and endoplasm. Right here is ectoplasm, the clear area is more hyaline, and then endoplasm is more granular, it's inside, and then way inside is cytoplasm. And here are the vacuoles, nucleus, and then contractile vacuole. We'll talk about all of that. Okay, here's in the lab, so you can see the cell membrane is a creamish color. Here is the blue is ectoplasm, and the yellow is endoplasm, and the orange right here, all of this is cytoplasm. And in this model, you have ectoplasm, endoplasm right here, and cytoplasm. And these are contractile vacuole. Okay, and these are all of the food, the bacteria they ingested. This is a vesicle. Okay, I hope. This is a contractile vacuole. This is the nucleus. And these are all of the bacteria, all of the food they are ingesting by the pseudopod. This whole entire thing is pseudopod. This thing is pseudopod. This is a pseudopod. This is a pseudopod. This is a pseudopod, and so on and so Okay, excretion and osmoregulation. So let's talk about that a little bit. What happens by contractile vacuole? The contractile vacuole was more primitive one. When I say for the rest of semester, whenever I say primitive, it means that the organism was evolved, the system, whatever you have, was evolved first. And then more advanced, opposite of primitive is advanced. Advanced is organism that evolved later on, or the system, whatever it is. So in this case, I'm talking about contractile vacuole. Contractile vacuole, when, let's go back right here. So these organisms, these amoeba, I cannot say animals. It was slip of a tongue, please forgive me. These are not animals, okay? So these organisms, water constantly goes inside of these organisms because of the osmosis. They are in fresh water, okay, number one, they're not in salt water. But again, there are more granules inside of the animals than outside of the animal. So the inside of the animal is hypertonic, outside of the animal is hypotonic. So water keeps going in. When water goes in, if these animals do not excrete them out, what will happen is the animals, this organism, again, forgive me, these Protista will burst open, right? So they have a system in place to get rid of this excess water. Here they are, some vesicles. I showed it to you before. There are vesicles right here, right here, right here. They grab the water that keeps coming in from outside, and these vesicles move and they bring the water into the contractile vacuole. Contractile vacuole then moves and attaches to the cell membrane and then opens up and excrete the water out. You know, that contractile vacuole is made up of phospholipid. Cell membrane is made up of phospholipid. So when they attach to each other, just like you learned in biology 1, the vesicles are, last exam, the neurotransmitters between presynaptic neuron and postsynaptic neuron, right? Between presynaptic neuron, postsynaptic neuron, neurotransmitters attached to the cell membrane and release, okay, the neurotransmitters. Same thing here, okay? With the difference that the contractile vacuole, it's not absorbed throughout the cell membrane. If it opens up, release the water, excess water, and then it closes down and move back a little bit. Okay, so there is intensive research going on whatever I'm talking about right now, the last 1 minute, 2 minutes of my talk. So, but briefly, you can ask a lot of questions that I don't know the answer, nobody knows the answer, okay, regarding this. So that is called the contractile vacuole, moving by contractile vacuole. Okay, small vesicles join the contractile vacuole, then contractile vacuole joins the cell membrane. The other one, a more complex one, more advanced, and it is complex. More advanced one, it means it was evolved recently. There are excretory pore leads to the outside and are surrounded by ampulla, and ampulla discharge the water to the outside. So as I said, water constantly coming in, right? Outside is hypotonic, inside is hypertonic, right? So when water comes in, instead of going to vesicles, they go into the endoplasmic reticulum system. All of this is endoplasmic reticulum right here, okay? From endoplasmic reticulum, then it goes to the ampulla. These are ampulla. And then the ampulla leads into contractile vacuole. One of the arms of the ampulla is called excretory pore, right here. When the ampulla squeeze, right, and squeeze the water out through that excretory pore, the water goes out. If you go online and look at the paramecium osmoregulations, go to YouTube. It's very interesting. You see, usually these organisms have two. So one becomes full of water and excrete the water out, and the other one becomes big. And they go like that. It's amazing. When I saw the video, I said, oh, wow. It is just like that. So one squeezing the water out, and the other one is becoming constantly, is becoming full of water. Then this one squeezes the water out, and this one becomes full of the water. So the name of that system, if you would, is most likely in a, it is the ciliated organisms and through excretory pore, through the ampulla and excretory pore. That's more right here. So of course, these are food vacuoles. They are not water vesicles. Micronucleus, they are food vacuoles. But you can see this one is filling up with water, and the water goes to the contractile vacuole. This is a contractile vacuole. These are ampulla. They did not show you the endoplasmic reticulum system in here. Okay. Nutrition, autotroph, it means photosynthesis. There are some that they do photosynthesis. You will see one in this class. Two, actually. Three. I'm sorry. You see three. And then heterotroph, it means heterotroph. It means hetero. It menes different, as you know. Auto, it means self. Heterotroph, trophim, it means food. Heterotroph, it means they obtain their food from the environment, like us. You and I are heterotroph. We are not autotroph. Okay. I'm sorry to break the news to you. And then the heterotrophs, they do either through phagocytosis, cell eating, by lysosomes. All of that was done in biology 1, food vacuoles, exocytosis, the waste material goes out, and then cilia have a cytoproc for expulsion of the waste. Pinocytosis is cell drinking pinocytosis. These are all, we discussed that in Bio 1. Fission, okay, we did not discuss that in Bio 1, so let's talk about it. Cell multiplication, well, we did talk about, I talked about fission in Bio 1, that is for bacteria. But these guys, the term fission, not fusion, fission, the term fission is also applied to Protista when a cell multiplication process, when more individuals are produced. It's pretty much mitosis. It's equivalent to mitosis. Fission is equivalent almost to mitosis. So binary fission, specific type of fission is called binary fission. Two identical individuals are produced. So when you have one protist and divide into two, okay, that is called binary fission. Budding, if you have, we've seen it in the lab on the microscope with some of the ciliated organisms, you have a huge organism and a small one bud off of it, right? And then it comes, this one becomes huge later on. Okay, it's called budding. Same, multiple fission. Then there are two types of multiple fission, sporogeny and schizogeny. I will give you the definition now, but when we get to malaria, I will elaborate on those a little bit more on these two, sporogeny and schizogeny. Development of spores or sporozoites within the oocyst. Oocyst, it means when the microgamete and macrogamete come together, okay? Almost like human fertilization, sperm and egg come together. So these guys, they don't call them sperm and egg, they call them macrogamete and microgamete. Macro, it means big, micro, it means small. So the microgamete and macrogamete come together, they don't call it oocyst, they don't call it zygote, they call it oocyst. Okay, it's different than cyst, and we'll discuss that. Schizogeny is when cytokinesis occurs preceded by several nuclear division. You remember in Bio 1, we talked about that when one cell divides and then the nucleus divides, mitosis, and then the cytoplasm divides. When the cytoplasm divides, it's called cytokinesis. Now we have a different type of multiplication here. The nucleus divides first, many of them, and then cytokinesis occurs, like that. I hope I'm making some sense. That is called schizogeny, okay? We'll come back to it. Forms and functions, there are two syngamete types of gamete fertilization, fertilization of one gamete by another one. Autogamy, gametes nuclei form by meiosis and fuse to form a zygote inside the parent organisms. And then of course, you have conjugations when two organisms come together and exchange nuclear material, and then they go separate, and then each one of them divide, which increase the genetic variability. As you know, genetic variability is advantageous. It's a good thing in life. Gamete nuclei exchange between paired organisms like the paramecium. Form and function is, okay, very good. Encystment and excisment, make sure you know it, okay? Because either I ask essay question from it, or there are gonna be several multiple choice questions from it. So, encystment, let's talk about encystment. If there are some Protista in a pond, in some kind of environment, and the pond dries up, or the environment gets cold, these organisms, they undergo encystment. And what does that mean? It means they form a cyst. And that cyst can stay in the environment for a long time, until rain comes, until the temperature goes back to normal. Okay, in some cases until another host ingests it, goes into the digestive tract of the host. So all of that is called encystment. And then when the rain comes, or the temperature goes back to normal, or when I ingest some cysts, they come out of cysts and they multiply and multiply and multiply by fission, binary fission is called excisment. When they come out of cyst, it's called excisment. Okay, excisment. So here we go. Unicellular forms are amazingly successful in extremely harsh conditions due to various survival mechanisms, and encystment related to the ability to form cysts, where dormant forms, okay. And then when they form a cyst, it's a dormant. It means they do not multiply. They do not do metabolism, much of a metabolism. They're just very dormant, almost, do not use that during exam or quiz, almost like hibernation, okay? But it's not hibernation. You're talking about single cell organisms, okay? That's why they give it the term encystment, or forming a cyst. So they become dormant, shut down metabolism, and have resistance to external covering secreted by Golgi apparatus. And but this does not happen in paramecium. Paramecium do not undergo encystment. Excisment escape from the cyst when environment conditions are favorable. Very good. Important for parasites forms to survive the transfer from one host to another host. Yes, you will see that. Okay, this is pretty much all of the organisms we study. I'm not gonna, oh, come on. I'm not gonna go over every single one of them, but I will, this is the most classification. If you're watching like 2 years ago, 3 years ago videos, and the classification is not like this, that is not acceptable. This is the classification I would like to go to by, and then please pay attention to it. I don't know how long I've been talking, but let's go ahead and do another 10-15 minutes, and then I'll call it off. At least we cover the next phylum, which is phylum Euglenozoa. Euglenozoa, class Euglenidia. So they are class, in the past, I did not hold you responsible, but whatever phylum I'm mentioning, and I'm mentioning the claim of the class, you should know the name of the class. It's not that difficult. It's just a few classes here and there you should know. Okay, so make sure you know the name of the classes as well in this exam, for this exam material. So phylum Euglenozoa, class Euglenidia, some species with light sensitive stigma. So light sensitive, it means they detect light. They cannot see, but they have stigma that can detect light, And guess what they need that light for? Photosynthesis. Okay, so, and they have chloroplast, the name of the organism Euglena, but not all of the members of Euglenozoa have chloroplast. Euglena does. Euglena has chloroplast. They have light sensitive structures called stigma. And then the other class is class Trypanosoma TDA. Okay, the trypanosomas and Leishmania belong to this one. So two classes in this phylum you should know. Okay, and they are very simple. Euglenozoa and then Trypanosoma TDA. They're all parasitic. When they are parasitic, it means they do not have chloroplast. They do not have stigma. Okay. Leishmania and Trypanosoma, there are two species that you should know, Leishmania and Trypanosoma. Let's talk about the Euglena. Remember from this one, the Euglena belongs to class Euglenozoa. So Euglena, some structures in Euglena, the pellicle, kinetosome, contractile vacuole, stigma to locate light, chloroplast, paramylon bodies, reproductive, and right here, oh, do I have any more or this is it? This is it. Okay, I do not have any more. We do have microscopic slide of these in the lab, and you should look at it. Here is a flagellum, of course they have. Here is an eye spot, so they detect the light, but they cannot see. Okay, second flagellum, they have another flagellum in here, so they have two sets of nucleus and plasma membrane, protein bodies in here. Where is paramylon bodies? Paramylon bodies, they are deposits of starch. There are starch deposits in here for the cells when they do photosynthesis. They do have chloroplast, and they do make glucose molecules. Okay, so when they make glucose molecules, they're stored in paramylon bodies. They do have contractile vacuole. They have a light detector. They did not mention the pellicle. Pellicle is a protein coat outside of the animal right here, which is called a pellicle. Okay, kinetosome, they are at the base, they have DNA, and again, they're under intensive research. They are right here at the base of the flagellum. So the function really, we don't know. I guess I used to know it when I was in college, but I'm forgetting now. In Protista, in Trypanosoma, I didn't know, but anyhow. So the class Trypanosoma TDA, the first organism is Trypanosoma. See, I got rid of the lines between the words. See that? I'm becoming better a little bit. So there is no word, there is no line between them. Trypanosoma brucei rhodensiense. Rhodensiense is found in Rhodesia, the country Rhodesia, in that area. And if a person is infected with it, it can cause death. And what happens with this parasite, the common name for this parasite is called sleeping sickness. Okay, so one of my professor, he went to Africa, or maybe he saw videos of it. He said people walk on the street and all of a sudden they fall asleep. They go in the corner, they go on the side of the street, they fall asleep, and after a few minutes, they get up, they walk away, they go to their, continue with their own life on. After a period of time, okay, again, life, it depends from the time they become infected until the time they die, it causes death. Okay, so from the time of infection until the death, they go into little short comas, sleep, and they get up and they move. Okay, so the other one is Trypanosoma brucei gambiense, the country Gambia, and the death is not so early on. Later on, it takes a little bit longer for the person to die, and it's not as in biology there is a term virulent as rhodensiense. Rhodensiense is more virulent. Then you have Trypanosoma brucei. Trypanosoma brucei is found in animals, in cattle, in elephants, in giraffes. That's what this is primarily in Africa. It can be found in parts of Middle East as well, in Asia, Africa. So the Asian parts that I'm familiar with, it is in Middle East. And then is transmitted by tsetse fly. The name of the genus, the common name is tsetse fly. The name of the genus is Glossina. At the end of semester, when we talk about the arthropoda, you will see that you have a microscopic slide of Glossina. But right now, do not worry about it. Just know that tsetse fly is a common name, and Glossina is the name of the genus that you should know. It transmits from human to human, from animal to human. So the fly goes take a blood meal from one person and transfer it to another person, take a blood meal from one animal and transfer it to human. So it is possible that we get it from other animals as well. The other one is Trypanosoma cruzi that is found in South and Central America. And there are a few cases of them reported from southern states of the United States, from very southern portion of the southern states of the United States. So, and it's transmitted by the kissing bug, Trichomania, and it's the name of the disease is Chagas disease. You know, the sleeping sickness and Chagas disease, they have some similarities neurologically. They have those similarities, but the Chagas disease patients, they do have some neurological problems because sleeping is a neurological problem that is caused by these organisms. But Chagas disease also have some heart problems. The patients have some heart problems, okay? And that's a big difference between Chagas disease and sleeping sickness. Trypanosoma cruzi, again, is you should know the locations. You should know Trypanosoma cruzi occurs in South and Central America, and Trypanosoma brucei happens in Africa and Middle East. And of course, they're transmitted by kissing bug and transmitted by tsetse fly. You should know everything there. The next organism in this class Trypanosoma TDA, is Leishmania. And in liver and spleen, Leishmania donovani, I did not give you the name of species. You should know the name of the species as well. Leishmania donovani, it's in your textbook. And what happens in liver and spleen is because enlargement of liver and spleen. We have 3 different types. I only mention 2 of them for you of Leishmania. We have visceral leishmaniasis, which is again spleen and liver becomes enlarged, and we have cutaneous, the face, and I'll show you some pictures, leishmaniasis. Okay, the name of the disease is leishmaniasis. So we have 2, and then we have the 3rd one, which I didn't mention. It is transmitted by sandfly. The name of the organism is sandfly. Again, Leishmania is found in almost all over the world, cosmopolitan, but mainly in Africa and South America, Central America, Asia, not Europe, not Australia, not much. It cannot be there. Here is the spleen and accumulation of blood. It's enlarged. And if you look at it, and of course, this is an autopsy I have done on this person, and the spleen is huge because of, and liver is also is more than its normal size. And it's because of the trypanosome. I'm sorry, because of the Leishmania. Here, you think this person is so skinny. Why? They have a huge belly. Okay, the reason for that is because they are infected with Leishmania. Okay, so Leishmania caused the huge. Here is a cutaneous leishmaniasis. This person, after surgery and medication and so on and so forth, went back to semi kind of, of course, he was disfigured, a semi kind of normal life, but they've done surgeries and then medications, and then that's a prime examples of a cutaneous leishmaniasis. So you saw both visceral leishmaniasis and cutaneous leishmaniasis. Okay, let's stop right here. I don't want to bore you guys. So we've stopped phylum Amoebozoa, and then make sure you know your diseases. Before I move on for the exam, it is important that you know the location of these diseases. When I say cosmopolitan, it means all over the earth. That's what the term cosmopolitan means. It means it can happen all over the earth. But when I say it's in Africa, or it's in Asia, or it's in this South Central America, then you should know that too. And the signs and symptoms, the name of the disease, and so on and so forth. Yeah, all of these you should be familiar with. All right, guys. So I will end, first stop recording.